from grade 12s, it's your time to shine. We are here. It's exam revision. You know, you're writing your finals tomorrow, and luckily we have good old Mr. Aslam here to take you through for life sciences. So you're here with me, Megs, and Aslam. How have you been?
Megs, hey, what can I say? What can I say? It's been a life science day today, and it's going to continue
He's just
through the night.
rushing, helping kids, going there. So I'm actually surprised he made it on time. He got you at like 3:20. So I'm so proud.
Just making it in time. We're doing revision of paper 1. Guys, you're writing paper 1 tomorrow, so we are just about paper 1 today.
Just about paper 1. So before we get into paper 1, let me tell you what's going on here at the Mindset Studios. You know about it, you've heard about it, we've said it so many times, guys. We are here doing the Learn Extra Exam Revision Marathon Competition, because you know this is a marathon. The finish line is how many weeks away? Like 2?
Wait,
Maybe
to the end of exams?
Yeah, like
it's about a week and a half.
Week and a half. So you only have a week and a half left of this marathon, and that is fantastic. So what's up for grabs is this Galaxy Note 10. Take a good look at it, Mindsetters. Look at that in all its glory. Proudly sponsored by Samsung. So how you enter the competition? Simple, it's easy. I post the notes on for you onto the page, the link. All you have to do is check out the race number at the bottom, and there's another link that you enter the competition with. But no stress, no worry, everything will be posted on for you just from me. So that's all you need to know. Before the end of this first ad break, I'm going to tell you about Curio and what you need to know, especially for you guys. So I think we should get into it because you guys have a lot to learn in an hour and a half. So good luck, and I'll be chatting to you on the Facebook page, and that is facebook.com/Learn Extra with an X, and Twitter, which is @LearnExtra. Big shout out to Batoni, all working. Love you guys, and see you guys tomorrow. So I think we should get going.
Thanks, Megs. Guys, remember the important thing is you must post your questions on the page. The more questions you post on the page, the more interactive we become, and then we are answering your questions, not our questions. Paper 1, just to remind you again, guys, DNA, RNA, protein synthesis, meiosis, genetics. In all 3 of those first topics, biotechnology. That means how life science or biology is mingling or is tangling, if you want to call it, with technology to make life better on Earth. Okay? So across the board, DNA fingerprinting, genetics, artificial selection, genetically modified organisms, meiosis, and different things around the chromosomes, all of that, and how that helps us in what we do. Then we move on to that, by the way, those 3 chapters are 60% of your work, and the remaining 40% comes from evolution, which is broken down to Darwin, the early theories. That means Charles Darwin, Erasmus Darwin, and Wallace and Lamarck, and the differences between Lamarck and Darwin, Charles Darwin especially. And then we move on to natural selection, speciation, evidence for evolution, and then we move on to human evolution and the branches there. That is what you need to know for paper 1. Please, 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 prepare for what is there. The exam guideline tells you these are the items that are in paper 1. If you're not learning these topics, then you are learning the wrong content. So let's get straight into some multiple choice questions first. This is adapted, by the way, look at the date there. It's adapted from the IEB paper, November 2013, guys, that was written this year. So this is fresh off the press, just at Mindset for you, so that you can go through these questions. Now, this paper was written in this week, by the way. So let's look at some of the multiple choice questions. The first one says, the term applied to the treatment of a genetic disease. To the treatment, again, underline the key words. By, this should be by, replacing a faulty gene. So we're replacing a faulty gene is known as cloning. Cloning, making a copy, so no, can't be that one. Gene therapy, disease, gene therapy, rings a bell. Karyotyping, what is karyotyping? Is the mapping of all the chromosomes in an organism. So that's out. Artificial selection, what is that again? Artificial selection is when man chooses desirable features in different organisms of the same species, and he tries to mate those organisms in the hope that their offspring will show that desirable characteristics. So now that's not the one. So we're left with gene therapy. And look how nicely it ties up. Treatment of a genetic disease by replacing a faulty gene. So gene therapy, curing, therapy, fixing, some sort of help. So that's how we do it. Notice the elimination process. We know these three things. This one here may be a gray area, so therefore we knock that off. By the way, I'm hoping you're following what I'm doing here. We're not just giving the answer to the multiple choice, but I've given you more or less an idea of all three of these other things that we are talking about. So those are, in their own right, some form of definition, if you want to call it that. But I won't say definition to the core, but more an explanation of what's happening there. Part of the coding section of a DNA chain is represented by the diagram below. There's a diagram below. Which of the following changes would take place in the amino acid chain being formed if the mutation below has occurred? So they're telling you, this is the DNA chain before the mutation. This is the DNA chain after the mutation. What has happened? It was AGA, TCG, AGT. Remember, we always work in threes. This particular A has been replaced by that C. Everything else thereafter is the same. You can see that clearly. Firstly, before we go into that, what type of mutation is this? Where only one nitrogenous base has been swapped by or replaced by another. We call this point mutation. If for some reason we inserted something here, we inserted a C here. Now what would happen? We would have AGA, AGA, then you'd start CTC, GAG. Can you see what has happened in that instance? Everything after that insertion has moved. So we call that type of a mutation a frame shift mutation. Why? Because the whole frame has shifted. Even if we delete something, for some reason we take that guy out. Now it's going to be AGA, TGA, GT. So again, whatever happens there, whatever happens thereafter would also be a change across the board there, frame shift. That's background knowledge on other things. So what I'm trying to tell you guys, just looking at a multiple choice question, we can learn so much other things around it. So there we have explained point and frame shift. But now they're saying, what will happen to the amino acid? The amino acid chain would be shorter than expected. There's a DNA chain, and for every 3 DNA codes, there would be 1 amino acid. So 1, 2, 3, 1, 2, 3. Would it be shorter? Nah, that's out. The amino acid sequence would remain unchanged. Would it remain unchanged? We've made some change. How can a change make no change? No such thing, so nah, kick that one out. 1 amino acid in the polypeptide would change. Possible, yeah? The amino acid chain would not be made. Nah, amino acid chain can carry on. So you're left with C. The answer is that 1 amino acid. Why? Because we only change one of these base triplets. And therefore, this will inform, because of that, in this place here, instead of, we would have had here messenger RNA, A, U, C, U, UCU. Now we're going to have UCG. And this messenger RNA would have coded for a particular transfer RNA, which would have been AGA. And now it's AGC. And these transfer RNA, each one of them, would have coded for a different amino acid. Different transfer RNAs code for different amino acids. So this is how it gets it. Again, guys, it's not about the answer. It is why that one is correct. Why is C correct? Get to understanding the work, you know. I've seen somebody studying recently, my daughter, I must say it on TV also. And when she's reading from the textbook, Megan,
Yeah.
the textbook says, as you have learned before, So she reads, as you have learned before, you wasted 2 seconds, precious seconds,
Yeah.
which you're not going to use again. Don't learn like that.
No.
Learn to understand.
That's
if you
the
understand,
best way.
you're going to remember.
Yes.
Because nobody can take that away after you understand. You will forget a few facts, but the broad concept you can't forget because you understood it. If you swat it, you can forget the whole paragraph or a whole section for an essay. Gone. So please think about that. Understand your work. Answer questions 1.3 to 1.4 with reference to diagrams 1, 2, 4 below. So they're giving us 4 diagrams. The diagrams show a single pair of homologous chromosomes in various stages of meiosis 1 and 2. So there's our diagrams. Something's happening there in 1, 2, 3, and 4. They're talking of just one pair of homologous chromosomes. So which of the following combinations is the correct sequence for meiosis? First of all, let's look at this diagram. Again, remember, you do not have to know the names of the phases, but you need to know what happens in meiosis 1, meiosis 2, what happens first, what happens second, third, fourth. The sequence you need to know. And where do we start? We start with the homologous chromosomes lying next to each other. So let's look at any of these diagrams where the homologous chromosomes are lying next to each other. It's lying there, not there. These are not homologous chromosomes anymore lying next to each other. Here they are, and here they are, and something is happening there. Okay, so any answer starting with 1 and 2 is incorrect because that's not how meiosis starts. So there's that one, we've got to take it out. Now we've got to go to 4 and 3 because most of them start with 4 and 3, 4 and 3, 3 and 4. Okay? So let's look at these two. What is happening here? Very important process in meiosis, and that is crossing over. Now, crossing over occurs after the chromosomes become visible and they lie next to each other. So we would have to start with 3. And the correct answer would be 3412A. This one is wrong because you can see clearly 2 cannot take place before 1. Here the chromosomes are pulling apart. Here chromatids are pulling apart. So this one we could have kicked out anyway. Okay? Now we have to decide between these two. And there we could come up with it. Now you can argue later if these things were in the middle and this is metaphases, but you cannot see them in the middle. There's no way they're showing. They're just showing that the chromosomes are visible and they're coming together. And thereafter, they cannot look like this after they've crossed over. They would be looking different if they have crossed over already. Which of the numbered stages in the previous diagram represents the chromosome undergoing reduction division? Reduction division means the chromosome number is reduced. So which of the numbered stages in the previous diagram represents that one? Is it 1, 2, 3, or 4? We said this is crossing over and this is belonging to that, so it can't be that one. This is in the second stage. It can only be this one here where the chromosomes are pulling apart. Reduction means the chromosome number becomes half. And what's happening here? They were together. They're being pulled to opposite poles. So half is going to one side, half is going to the other side. Yes, you may argue that it's happening here as well. But here, it has already been halved. This one here is the chromatids that are moving. So we're going to have the same number moving. Here, one chromosome is moving that way, one is moving that way. Here, 2 chromatids are moving in either way there. So there's 2 in either way. There were 2, there's still 2. Okay? There were 2 chromosomes. Now there are 2 chromatids on either side. So it will be number 1A. Okay. This one here we're going to look at here. Which of the following statements best describes the relationship as shown by the graphs? When the temperatures were high, the mammals increased in size. Here's the mammal size going that way. Here's temperature. What's happening here? The temperature is coming down. What's happening here? The mammal size increased. So it can't be this one. I'm not even reading the instructions, just by looking at the graph. So it can't be that one. Larger animals are not easily caught by predators, and so they are not likely. Is that related to temperature? No. A cooling climate results in all the mammals only passing on large genes to their offspring, A decrease in temperature shows a corresponding increase in the average size of mammals. Let's see. A decrease in temperature shows a corresponding increase in the mammal structure. That is shown there. After that, it doesn't happen again. It's in that area there. So your answer is between these two here and that one. A cooling climate results in all the mammals only passing on large genes to their offspring. We do not have enough information to make their judgment about passing on genes, so we would go with D. That would be the correct answer, and that is the end of the multiple choice questions.
Woo! Aslam is flying through this and he's rocking it out. So guys, I want to tell you, grade 12s, especially for you, this is the Curio challenge that is given to us here at Mindset. So how does it work? You register on www.curio.co.za and you enter the passkey, which is LS10, not 12, 10. But I'll post it on the page for you, facebook.com/learnextra. We'll see you after the break. Welcome back, grade 12. So as we left before the break, I told you about Curio, and what I forgot to mention is that you win fantastic prizes. The person who answers the most questions on Curio the fastest gets 100 Rand airtime. I could use airtime. And also, if you enter a lot of times, so the more you enter, there's a big prize at the end, and that is a Galaxy or S3 Mini, proudly sponsored by Samsung. And don't forget, there's cash prizes, so enter, do the Curio challenge. It's only for your benefit. So while during the break, Aslam and I were looking at questions. A caller.
Wow.
Oh, this is new. Can we talk about it? What's going on? James.
Through, James.
Okay.
Who are we talking to?
Yeah?
Penny?
Yeah? I can't hear her. Hi, caller. Oh, well. Okay. Chanel Govender says, are we required to learn Mendel's laws?
Mendel's laws, definitely you need to know Mendel's laws. And by the way, guys, not just on the theoretical side, if you know Mendel's law, you will be able to answer the questions, because the way genetics is done will follow what Mendel has said.
Okay, and one more just before we carry on. Themba Maleka says, what is the difference between gene therapy and gene testing?
Gene therapy? Come on, guys. You don't need to ask Aslam that or Megan. Gene therapy and gene testing. Gene therapy, therapeutic, fixing, where it is used in the treatment, testing, or what we would use for DNA fingerprinting when we're testing the genes, or we're checking the genes that we are getting from people we're testing. So the one is the testing, we're taking out the DNA, like in the DNA fingerprinting, we're doing some testing there to check the genetic makeup. And the therapy is when we're using genes to fix problems.
Okay, and Maureen and Irene are the twins. Say, Mr. Aslam, we are so excited to see you. Thank you for the revision, and we love you. So,
Maureen and Irene, hi, and hi again, because there are two of them, eh? I must say hi once.
Hi.
Good. Fraternal twins of ours, eh?
Yes.
Okay, let's move on. The following diagram shows, again from IEB, shows part of a cellular process. Now, guys, this is one way in which this particular section of the work is tested. And you'll notice that when we're going through the questions now, the multiple choice was all over the place. But what we're going to do now, we're going to go through sections. And hopefully, we're going to cover a question or questions on each of these sections as we go along. All right? So this section, this is one way in which it is tested. And we're going to see others as we go along. So we're saying that this is a diagram that's showing something. So they're showing you something here happening. Something happening there, and something forming there, and that thing there moving in that direction. Name the specific process. I was just chatting to John earlier, and I said I was discussing a question like this with my learners, and I didn't read that word specific when I was discussing it with them, and I had to come back after I saw the next question. So please, read the question properly the first time. The specific process. So not talking of the general process, there is a part of a process shown here. Obviously by now we know that this is protein synthesis, and this part of protein synthesis where the DNA is unwinding and this molecule is forming there, molecule B, that would be transcription. Okay? What is the end result of the whole process in the cell cytoplasm? The end result, the purpose, what is this again? Protein synthesis to make proteins. So the end result will be the formation of a protein. A and B are both nucleic acids found in living cells. Draw a table to illustrate one difference in structure and one difference in function between the two types of nucleic acid, A and B, shown above. So we're going to go back. There's A, there's B. Now, first and foremost, nobody asks you to name these two molecules. They only ask you to tabulate the difference. If you're not sure which one is which, if you didn't know that this is DNA and this one is RNA, messenger RNA to be more specific, then don't write DNA and mRNA. For example, if you label this one as messenger RNA and you label that one as DNA, now if you put it in a table, that means your whole table is incorrect because you got the wrong things under the wrong heading, because you've identified it incorrectly. Use the letters that are provided. They gave you the letters there, A and B. So you use the same letters, and that's A and B, okay? So we're going to draw a little table there. We say A and B. In a question where they're asking you to tabulate, the heading does not matter, like this. When they give you data in an experiment investigation, and then they tell you to show your results in the form of a table, in that case, you must have a heading on the table with both variables, just like you would in a graph. Okay? But in this one here, you notice there's 5 marks. There are one difference in structure and one difference in function. So there's two differences. So 2 times 2 will give you 4 plus 1 for drawing the table. So the one as A we saw as DNA, I think. Yeah, DNA. So in terms of structure, double strand, single strand. Sugar, which is deoxyribose. Remember, you're only going to get credit for one difference. We're giving the others. And here we have a ribose sugar. Then you go further. In DNA, we have one of the nitrogenous bases are thymine, or we can name them all A, T, G, C. And in RNA, we have uracil in its place, or we can say A, U, G, C. But you must name them. Obviously, you can't just write it just the way I've done it there. So that's the story there. And in terms of structure, some textbooks also say that DNA is a longer strand and RNA is a shorter strand. So those are the 3 major structural difference. In terms of function, the easy one to know is the one of messenger RNA. Messenger RNA's function is straightforward. It copies the code from DNA and takes it to the cytoplasm. More specifically to the ribosome. We'll come to that just now. Whereas DNA has many functions. Number 1, it contains hereditary information. In other words, information about our characteristics that can be transferred to offspring. All right? And it is also involved in protein synthesis. This one is also there, but we don't use that as a difference there, because you say protein synthesis, you can't use it because messenger RNA is also involved in protein synthesis. So those are the 2 important things that we can use, and any other function of DNA that is different from protein synthesis. I'm hoping that we are clear with that one. Megan, you okay?
Yeah, I'm good. How are you?
I'm fine, I'm fine.
He's teaching me while I'm sitting here, so.
Good. Describe the role of molecule B in the whole process mentioned in question 2.2 above. Question 2.2 is this protein synthesis here that we are talking about in the whole process, okay? What's the end of the whole process? Good. So they want to know in the whole process, not only in transcription, but messenger RNA's role is transcription. So messenger RNA is involved in transcription. So you're going to explain transcription. I'm not going to write the whole story there. Transcription is when messenger RNA copies the code from DNA using the bonds of the nitrogenous bases. In other words, A will combine with T, U will combine with A, etc., etc. G would C and C would G. And in that way, the messenger RNA forms, and it takes this code and it takes it to the cytoplasm, attaches itself to the ribosome, exposing the codons. And that's where its role stops. That's where its role will stop. So that's what you can say. And notice that they're not asking for too much there. They're only asking for 3 marks. And you needed to select information of the role of this molecule. You didn't have to explain the whole of transcription, because in between you can say DNA unwinds and so on, replication. There's no mark for that. There's no mark for saying that enzymes control this process, because they want to know the role of messenger RNA in this process. Are we clear with that? Good. Question 3. Right, they're giving you 2 partial sequences of DNA bases, one from a human, one from a cow. In the table below, the sequences in both humans and cows contain the gene to make up the protein insulin. Also provided is an anticodon chart that can be used to determine which amino acids are assembled to make the insulin protein in both cows and humans. Okay? So they're giving you the whole story. This is a different way in showing this question. Remember, this is that other level now. The one was the one I showed you now. Diagram questions, or explain protein synthesis under these headings, or the essay on protein synthesis. Those are the one way. The second way is when we use this nitrogenous bases to see whether you can match the DNA to the messenger RNA, the messenger RNA to the transfer RNA, and the DNA, the transfer RNA, and the messenger RNA to an amino acid. So this chart is giving you all of that, all these anticodon charts that giving you all these things here in between with all the sequence there. And here's the human DNA sequence, and here's the cow DNA sequence. You can see in some cases they're similar, and there are some differences. There's a difference there. That one is the same. There's a difference there. Same, difference there. Same, and totally different there. So there are some differences. So you say name the process that forms messenger RNA during protein synthesis. Now the other question informs that answer for us, and that would be transcription. It's during transcription that messenger RNA is formed. Notice we say that the function of messenger RNA is to transcribe, to copy the code. And we're saying also that messenger RNA forms during transcription. Give the messenger RNA strand that is formed from the base triplet number 7 in humans and cows. Base triplet number 7, yeah, this one, TGA, CGA. TGA, CGA. So we got to look for TGA, CGA. What's that? Human DNA sequence. All right? Anticodon, so the transfer RNA. Remember what you had to do here, but now you needed to be a bit sharp, because this is a DNA sequence, TGA, CGA. This is an anticodon chart. The anticodon is found on the transfer RNA. So we need to find out what's the transfer RNA for this guy. Let's just use another color so that you don't get confused. That would be A, that would be C, and that would be U. And this one would be G, C, U. All right? Now we need to go here and see where that is found in this chart. And from there, we would then use that to get, in humans, it's ACU and GCU in that case there. All right, we've already worked it out in the case there, what we've just said now. All right? Give the correct amino acid sequence for the base triplets 1, 4, and 5 in the cow DNA. And that would be proline, valine, threonine. Now what they're saying there, 1, 4, and 5. So you're going to go to 1, 4, and 5. You're going to do exactly what I've done here. You're going to go to this chart, and you're going to choose the amino acid that matches that transfer RNA. So that's what you're going to do. Okay? An error occurs during the formation of messenger RNA from human DNA. This results in cysteine in base triplet 6 being omitted. Important word. Very important here. Omitted, left out, deleted. All right? This is on cysteine in base triplet C from being out. So it should be cytosine actually. In cytosine, not cysteine. Cytosine. This results in cytosine from base triplet 6 from being left out. So there you have base triplet 6, 7, 8, 9. Base triplet 6 to 8 will now read as follows. AGT, TAA, GGT, AA. So what has happened? Let's go back to the chart. Here's number 6. So what has happened is that the C has been taken out, omitted. Okay? So now from there, what has happened? AGT, GAA, GGT, AA. Exactly what they're telling you here. Oh, we discussed this under the multiple choice question. We said there is a frame shift mutation. Okay? Mutation. Name the mutation. Well, I gave you the answer already. Name the mutation that occurs above frame shift. What effect will the above mutation will have on a human being? And let's just go through that answer quickly. Obviously, TRPM6 to 8 will code for a different sequence of amino acids. All these here will be replaced by serine, glutamic acid, and glycine. You'll have to take the codes and match them to your chart. And you're going to be specific about it. And at the end, first you're going to say that there's different stuff. At the end, a different protein from the original protein coded for will be formed. A protein that cannot function may be formed as well. Use evidence from the table to explain how cow insulin can be used in humans if human insulin is not available. And obviously, human and cow insulin are very similar. I told you this when we were doing that. Triplet 1 codes for the same amino acid in both proline and so on. Therefore, the proteins formed by each will be very similar. And in the past, we used animal insulin to treat people that were sugar diabetic. And we've moved away from that, from using genetic therapy, as you want to know, and we make our own insulin. I think I said too much.
It's too much. I don't think so. I think you learners out there are learning a whole lot because I see you guys are very active on the page, and this makes me super duper happy. So a big shout out, I hope I say your name right, to Fenyo Mosepele. He says, please shout out to me. Hope I said your name right, but don't stress, we'll be back after the break. And we're back, Mindsetters, grade 12s. You're writing life sciences tomorrow, and you're so lucky enough to have me and Aslam here for only you to answer all your questions that you're posting on Facebook as well as callers. Yo, that threw me off a little bit. When someone phoned in, I just stared into oblivion. Yes. She
called earlier and she wanted to know about essays. Guys, we're going to be dealing with essays a little later. Later.
He wants cats.
And we will then talk about the essays and how to answer essays. There's lots of you that are asking those questions on my own WhatsApp as well. Yeah. I'm getting a lot of queries about essay writing, so we will deal with that later.
Okay, awesome. Well, there's nothing left for me to say, so Aslam.
Megan, we had a lot of questions on evolution as well.
Okay, yes, yeah. I said
so we're going to take those. Please, guys, just bear with us. Follow the program. When we get to evolution, if we've answered your queries, then you're okay with it, give us a thumbs up. If we haven't, then repost your question in terms of then we can deal with it. I don't want to confuse the topics. We're dealing with a certain topic now and we'll come to other topics later.
Yeah, we can't jiggle and
just be there with me, guys.
All right.
This one here, the thing said, use the evidence on the table to explain how cow insulin can be used in humans. Just to recap that one. If you look at, and I showed you this as I go along, that the fact is that the human insulin and cow insulin sequence is very similar. And in that because of that reason, it can be used for humans. And like I said, we used animal insulin in the past before we learned how to manufacture human insulin using genetic technology. Okay? So those are some of the ways in which protein synthesis can be stuff. And we've dealt with also, by the way, we've also dealt with frame shift and point mutation. That is, remember, when you talk about mutations linked to protein synthesis, there's only two, and that's frame shift and point. And you need to show how the one affects. The one only affects one amino acid in a chain. The frame shift affects every single amino acid after the change, whether it was a deletion or an insertion. Everything after that. That is why we're saying frame shift. The frame has shifted. Okay? Good. Question 4, DNA fingerprinting. I'm not going to, all right, we'll have to read this. The diagram below represents DNA profiles of a family consisting of a mom, a dad, as well as 2 daughters and 2 sons. They gave the names there, one of whom has been adopted. Study the profiles and then answer the questions that follow. So we got Anne and Chris with a mother and a father. And then we've got the children, the 2 daughters and then the 2 sons. Okay? And then they gave you the genetic profile. Remember, the quality of the diagram may not be very clear, but it's enough to answer the questions for us. Okay? Give the name of the child that Anne and Chris adopted. Now, this one here, this DNA fingerprinting is different from that of a crime DNA. In a criminal case, the DNA generally matches one of the suspects. So it's an exact, so it's easy to see. That one looks the same as this one. But when it comes from parents to children, none of the children would have the same identical DNA of any of the parents or any of the siblings, because each one of us has a unique DNA, apart from identical twins. Not our twins, Maureen and company, they are fraternal twins.
Yeah.
So they have different DNA. But others, identical twins coming from the same egg and the same sperm, they have the same DNA. So what we have to do, again guys, it's not so complicated, it's a matching exercise. We've got to look at, here's Anne, go across, there's something from Anne there. Oops, let me just get this thing right. There's something from Anne there, but this one, yeah, there's something there. Then there's something from Anne there. There is something from Anne there. There, and there. And now, let's look at in terms of Chris. There's something of Chris there as well. There's something there of Chris. No, this is different from Chris, so we can't say anything there. Here, there is something of Chris there. There's quite a bit matching. Alex, any dots by Alex? No dots by Alex. So therefore, Alex would be the one that is adopted. Okay? Because none of his DNA strips match either one of the 2 parents. Okay? The second question says, which child, now listen to this question, Emma, Kelly, Sam or Alex was the offspring of Anne's first marriage and is not Chris's biological child. Notice why my homework was important. Can you see what I did? I didn't go into the answers. I did some homework, I did some matching, and I did this. You can do the same in your paper. Now what we see this one got pink. That is, I didn't deliberately choose pink and blue, but it worked out fine. Pink for Anne and blue for Chris. All right. Kelly does not have a blue in it, so there's no bands of hers that are matching Chris. So therefore, Kelly has to be that other child from the previous marriage. Give an explanation for your answer. I've given you the explanation already. She has DNA bands that match her mother's, but no DNA bands match Chris's. She has bands that are different to Chris. Now, here also they could have asked you for the explanation, and the explanation is none of Alex's DNA bands match either one of his parents. Explain one other use of DNA profiling. So they're saying here, we're looking at parenting or paternity and maternity in this case. What other reasons do we use DNA profiling? And I alluded to one of that earlier, and that is crime scene, solving crimes. Also, when we find human beings in a disaster, they cannot be recognized. We can then identify who they are, or in a murder scene where we find a body after many years, we don't know who this person is. DNA fingerprinting can help us trace back to that. DNA fingerprinting can also be used to diagnose certain disorders and also in the treatment of those disorders. So there's quite a bit of other reasons. So medical, criminal stories there, and this other story there as well. Good, we're now moving on to meiosis. So we've now covered the DNA part. We're moving on to meiosis. We'll start with a very easy one. Study the diagram below and answer the questions that follow. Name the process illustrated in the diagram. Lovely diagram showing you the process there. What process is that? Obvious, it is crossing over. It's not meiosis, it is crossing over. This process, not the cell division. The cell division is meiosis. This process is crossing over. Provide a label for the part marked Y, the part where the crossing over is taking place. That would be chiasma. If there's more than one, plural is chiasmata. Give one reason why the process mentioned in question 5.1 is important. One mark only, so you're just going to say it increases or promotes genetic variety. If they ask you or let's see the next question. What the next question is? Describe what process, what happens. All right, let's see. Let's do that one rather. Describe what happens during the process as shown. You're just going to describe it. Homologous chromosomes, bivalents, pair up. They lie next to each other. Each chromosome has 2 chromatids. The chromatids overlap or cross over. Genetic material from the one is exchanged with the other. Another process, after the process of crossing over takes place, the chromosomes are different from what they were original. Okay? That's all it's saying there. Another question they could ask you in this kind of type of question is to draw one of the 2 chromosomes after this thing has happened. Now, nobody asks you to draw it exactly like that. You could use your pen. For example, let's say we want to draw this chromosome here on the left-hand side. So let's keep our pen going in one direction with this one, there, and we got that happening there. Notice you don't have to draw the whole thing like that. And we still got that, and then we got that, and then we got a dent. After that, there is some change there. There, my pen won't join, but that's what we're saying. So whatever happens at that crossover after that, so that one is joined there, that one is joined to the other one. So that's how you join. The idea is to show crossing over has taken place. You must be able to label the different parts of the chromosome as well. Okay? Are we happy with that? Good. Where are we? Describe what happens there. We've given that already. So that's one question on meiosis. Another question on meiosis. The diagrams below show cells dividing during meiosis from free state. Give the names of the parts labeled B, D, and E. B is the structure shown there. Now notice, although that is the end of the cell, so we refer to it as the pole of the cell. The cell does not have a pole, a physical structure called the pole. That structure is the centriole. Okay? So that's the centriole. But because the diagram is not clear, it could also be pointing to the spindle fiber. But the one at the end is the centriole if the diagram is clear. Number D, these are the daughter cells. You must be able to say daughter cells or even haploid cells. And E is the chromatid. Those are the labels. How many chromosomes does cell 3 have? It has 4. Now here, this is where I have a problem as a teacher, an examiner, a marker, whatever. You know, we give a question like this, Megan.
Yeah.
And we ask them how many chromosomes here.
Hey, don't ask me
and they
that.
get it wrong.
I would.
But the thing is, it's a counting exercise.
Okay.
One chromosome is made up of 2 strands.
All right.
So how many things in this diagram you see that I have 2 strands? 1, 2, 3, 4.
4.
That's the answer. But maybe you are thinking there's something funny
More
about
into
it,
it, yeah.
and so on. Don't
tend
think
to do
that way.
that.
Just count this story. When they're asking you what is going to happen after, yes, now you're going to divide it by 2 because
There we
meiosis is reduction, division. Good? Explain whether cell 3 is haploid, N or 2N. We could either give it N or 2N, or we could say haploid or diploid, okay? So, what do you notice there? Are the chromosomes paired? Yes, they
Yes.
are. If they paired, they would be 2N or diploid. If they were single, then they would be haploid. Explain one reason why meiosis is important. And now they're asking you for the significance, guys. This is from the textbook, straightforward textbook stuff. First of all, genetic variety. That's one of the important things of meiosis. It brings about genetic variety. Number 2, it maintains the chromosome number from generation to generation. For example, we as humans have 46. Now imagine if meiosis did not take place, our sperms would have had 46, the egg would have 46, our children would have 92. And chromosomes are species specific. The number of chromosomes is particular in a species. And it's not about advancement in any way. For example, ferns have 300 chromosomes. We have 46, we have less. We consider ourselves smarter than ferns. I wonder sometimes if we are indeed. But nonetheless, so we are saying that meiosis doesn't allow that from happening. So reduction of chromosome number and the formation of gametes. Meiosis is important for the formation of gametes. It also brings about a variety we already spoken about, form gametes for sexual reproduction. Meiosis is also involved in plants, allows for alternation of generations. So the 3 main ones for me, genetic variety, chromosome number being constant, formation of gametes, number 4, number 3, and one last one, alternation of generations in plants. Good. We're moving. We are now moving to, all right, we're still with this one here, bit of meiosis again. Nice that we're having something else, problems in meiosis. Group of grade 12 learners did a survey about the occurrence of 3 genetic disorders in a small human population. The results are shown in the table below. Number of people in the population is 1,500. Results of the survey of the frequency of the genetic disorders in a population. There's it there. It's telling you that 9, 12, X in albinism without any disorder, 75%. Which disorder is most common in this community? And I love questions like this for you, not for me, but for you as a learner, because even if you didn't learn your work, you can read from the table. Huh?
And you still get marks. So.
Yeah, you're getting some marks just for reading and analyzing the table. Which disorder is most common in this community? It would obviously be, which one is just that one there? You've got to look at it. Aye, come on. It will be sickle cell anemia, 12%. Calculate the number of people with albinism, show all working. Again, come on guys, you all do maths, lit or arithmetic at least in your life. So you know, you must know for a percentage to be, a total of a percentage, 100. So we got 75, 12, 9. So what must you do? 100 minus, in brackets, 75 plus 12 plus 9. And that is how we show that. There, 100 minus 75 plus 12 plus 9 is equal to 4%. And that is 4%. But notice what they asked you. They asked you the number of people. Again, they're asking you for the number of people, not the percentage. So the first part was easy. We worked out the percentage. And they told us in the stem of the question that there were 1,500 people. So we work out 1,500 times 4%. And you don't need, you'll have to show that on a working out. But on a calculator, you're going to get your answer there. Give one reason why genetic counseling would be important for parents who are expecting a child that may have a disorder such as Down syndrome. Why is it important? It's important to let them be aware of the risks and responsibilities that we to deal with a child with Down syndrome. So first of all, I wouldn't go straight to that. I mean, that's harsh.
Yeah. You know, can I tell you a little secret about
Yes.
me? I was apparently supposed to be a Down syndrome child.
Oh, that's what was wrong with you. I should have known. I was wondering all these
Thank
days
you,
what's wrong with
Aslam.
you.
Thank you. I feel so hurt. Yeah. So I was supposed to be, but then they ran the second test, and that second test showed
But
that I
Megan,
was
what you have
normal.
brought out is such an important fact. Now, we will ask, what's the arguments against amniocentesis? Amniocentesis is the test that would have told Megan's parents that she's probably going to be Down syndrome. Wow. That's we don't know what value you've added to this thing.
Yes.
Now, what is the argument against? The parents may get depressed because they're thinking they're having a child with Down syndrome.
Mm-hmm.
And when the child is born, the child doesn't have Down syndrome. The same goes for knowing the sex of the baby before time. Let's say a couple want a boy, and the doctor says it's a girl. The mother may go into depression. The father may go into depression, going into anger. In certain cultures, you know, they want a boy, or the other way around. So those are the arguments against those things. So it's not always good. And if you believe in the book, in any book, I'm talking of religious book, then you will know that the ultimate architect is up there beyond our science and biology and maths and the rest of it. So you will have to do those type of moves. Thanks, Megan, for that valuable contribution.
You're welcome, Mr. Aslam.
Me, I wouldn't go with that first one first. I would
Yeah.
say help prepare parents to manage with such a child.
Yeah.
So make them know, look, this is a Down syndrome child. This is the symptoms that you're going to get in a Down syndrome child. And this is what you'll have to make changes in your home, maybe, and your lifestyle for that. And let me tell you, the way we've advanced now, Megan, a Down syndrome child goes to school even.
Yeah, of
and,
course.
we're getting people that are progressing in that way. I
great.
think we've overshot.
Yeah, I think
we've punished them a little bit,
time.
but that's fine. Let's give them a break.
Thanks, Aslam. Guys, quickly go grab a glass of water, and me and Aslam will be right back after the break. Welcome back, Mindsetters. So you're here at Mindset, learning more, learning extra with Aslam and myself. So I hope you guys are actually understanding. If you don't understand, Aslam and I go through it during the ad break while you guys are watching all the pretty things. We actually look at your questions and see what you're saying. So that is facebook.com /learn extra with an X, okay? And that's where to find us, post any questions, any concerns, and take the Curio Challenge, guys. You register www.curio.co.za, and then you type in like the little passcode, and that is LS10. So do them in the fastest time and quick. But we have a question here, Mr. Aslam, from Matsobeni Tefo says, what are mutagens? I think he didn't know how to spell it. And a few examples of them, please.
Okay, Matsubano.
Matsubani.
Well, mutagen,
Yeah.
coming from mutation, gen, something that causes a mutation. Anything that causes a mutation, just like carcinogen is anything that causes cancer. Okay, so a mutagen is anything that can cause a mutation, and that examples of that would be any chemical, smoking, smoke, pollutant, X-rays, a host of other things that can cause mutations are called mutagens. Good. We move on to genetics, and we're going to rush through the genetics, but the easy part we're going to rush through, and then you will understand why when we get to the evolution part. A blue-eyed woman marries a man who is heterozygous for brown eyes. So that already tells you a blue-eyed woman marries a man who is heterozygous for brown eyes. It already tells you if someone is heterozygous for brown eyes, that means brown eyes is dominant. You can't be heterozygous for brown eyes when brown eyes is recessive. Brown eyes, they're already telling you that brown eyes dominant over that and what have you. So they're telling you represent the genetic cross. Notice the move is a genetic diagram. No question will tell you draw a Punnett square or a line diagram, because both are acceptable in the exams. Both are accepted in the exams. You can draw a line diagram if you're comfortable. You can draw Punnett squares if you are comfortable. The important thing is to keep to the entire format. And let's look at that now. There's 6 marks given. Notice that this format doesn't change. We've got to start with saying P1 or parent, and there we will give the headings phenotype and genotype. Phenotype, we're saying one is blue eyes, one is brown eyes. If you got all that right, phenotype, blue eyes and brown eyes, you're going to get 1 mark there. Okay, then you're going to talk about this, you're going to get 1 mark, your genotype. This was homozygous because it's recessive, and that is heterozygous because they told you it's heterozygous. Then you're going to write meiosis, because meiosis then forms gametes. There's your gametes. And this one here is showing you the line diagram. The lines moved for some reason. They should have been there with these things here. In other words, this is what should have happened. There and that. Now see why I don't like line diagrams. They can be clumsy and they can be all over the place and you can make mistakes. So the Punnett square is actually neater and it's easier for you. Whatever is on the left, keep it on the left. Whatever is on the right, put it on top. That's my rule, not Mendel's law. Right, there you're going to do all of that. There's that one. There's that one. There's that one. And you can put it either way. And there you got that one there. Okay? And there you're going to get your marks there for the marks as you go along there. Notice you must have parents 1 and F1. That gives you 1 mark. You must have meiosis and fertilization in the right places to get the 1 mark. So any genetic cross out of 6 marks, you're going to get 2 marks at least if you do this format the way it is. Please consult your memos, consult our previous lessons and our previous notes. We have given you the format of genetics crosses. Also, if there are other questions where it says, show a Punnett square for 2 marks or 3 marks, then you don't have to follow the whole thing. But if they say show Punnett square and they have 6 marks, 6 marks is aligned to these 6 marks that are given here. So please make sure that you are doing that. That was a very easy one, so we're not going to go into the details. They went further with that one. Okay, there's the Punnett squares that came out there in the end. There's your Punnett square here. Normally it's in there. This is just to show you we could have did the same thing with a Punnett square as well. Good? I've drawn it for you in the diagram, right? Now this one is one I want to spend a little bit of time on. And I want to show you something very important here. They say Fabry's disease is a sex-linked disorder. Already they're telling you sex-linked. The minute you see sex-linked, you must know that you have to work with XX and XY. If they don't tell you sex-linked and they tell you it's hemophilia or color blindness, then you must know it's sex-linked because those two are prescribed in the syllabus. This one is not prescribed. Notice how much of information they gave you. They told you what Fabry's disease is, and they told you that it's a sex-linked. That's all they needed to tell you here, because the question is based on how you work it out. They're giving you the whole story there. The diagram below shows the occurrence of Fabry's disease. So this one incorporates a pedigree diagram and sex-linked. Okay? So they're telling you here, this is a normal male, there, affected male, normal female. That's number one. So if we know this, on your question paper, take your pencil. By the males, write down XY, 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 XY. And the females, X, 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 X. So you know when you're answering this question how to go about doing it. Okay, so now, but they're telling you that Fabry's disease is caused by a recessive gene. So the ones that have it will have a small f. So we go to the males. This male is affected male here, so that will be small f. This one is affected, small f. Notice in the males, because the Y chromosome does not carry many genes, we do not have a counterpart. And that is why more males will suffer from a sex-linked disease than females. Because the females have another X where they can have the normal gene as well. That one is clear. That one is a small f again. That one is clear. If it's clear, it's easy. You're going to put the capital F, obvious, isn't it? Because they are normal. Now the problem comes in the females. This female, because they have two X's, they can be XF, XF, capital letters, or XF, capital XF, small, heterozygous or homozygous for it. So this is the problem now. What do we put there? How do we do this? We go below. Here's the parents. In this generation, let's look at the kids. This one got capital F, so you must have got it somewhere from the father or mother or whatever. Okay? But look at these ones. They got a small f on the X. They all got, this one got a capital X, F on the X. Now, where did these kids get the small f from? Could they have got it from the father? No. They can't get it from the father. Why? Because if they got an X from the father, they wouldn't be boys, they would be girls. Because the mother can only give X. She got one X, she got another one X, she can only give X or X. The father can give X or Y. If he gives you Y, if he gives the fertilized egg a Y chromosome, then that would be a boy. Okay? So now, where did they get the small f from? They got it from the mother. So we give her the small f. But we know that she's clear, so she's normal, so she must have the capital F as well. So the same goes for this female here because she has the same problem here. X, capital F, X, F. And that is how we work. Notice the homework I'm doing. I haven't even looked at the questions. I've done all of that. Now I come to the questions. Give the genotypes of the individuals number 2 and 3. We've worked it out already. And there you got that, as we have spoken there. How many unaffected individuals in the family are carriers of the defective genes? Can males be carriers? No. A male is either affected or not affected. So already all the males are out. You're left with 2. Are they carriers? Yes, both are carriers because they have the gene for it. So 2 are carriers. I have enough. 2 there. Draw a genetic diagram to show the chances. Now, notice here, this is important. Notice that the way that you set out your answer does not change. It stays the same. Only you're changing your letters X to the normal letters. And when you're doing blood groups, then you're going to use the letters IA, IB, or I to work out. You're using the same things, you're substituting it in a diagram, and you don't have problems with that. I'm not going to the answer there because it's straightforward. You just have to work it out there. If you're a genetic counselor, discuss 2 possible options you could possibly to give to these people that show that now they got Fabry's disease. Now, whether it's Fabry's disease, whether it's Down syndrome, whether it is color blindness,
Thank you very
whether
much.
it is color blindness or whatever else, it doesn't make a difference. The counselor will do the same thing. And let's see what the counselor should do. There's it. You see, the counselor says you can have the child, but prepare yourself to deal with it, or you can choose to abort the baby and try again later. That's not a nice thing to tell a couple, but nonetheless, notice the counselor's work is not to tell you what to do. The counselor will expose you to the options. I would like to be a psychologist, you know, Megan.
Really?
They do nothing. And they get paid
To
mega bucks.
give
You go to them,
advice.
let's give you an example.
Yeah.
You come to me with a problem, just tell me any problem.
Let's think. A
You had
problem.
a bad day at school.
Yes.
Right.
I was bullied.
Okay, and how did that make you feel?
Very sad.
Hmm. Why did it make you sad?
And
Can
then
you see what I'm
give
saying?
all the answers
See,
anyway.
you're giving all the answers and you're paying the guy for all your
ridiculous.
answers. What a waste of money. Hey, it's not a waste of money. Counselors are experts with what they do. Guys, with that,
Ad break. Time for an ad break, guys. Don't go anywhere. Facebook.com/Learn Extra for all your questions. See you now. Welcome back, guys. So I think it's Bupelo or Boypelo. She's been telling me to give her a shout out and I haven't had time. So now I'm giving you the shout out. Hi to you and thank you for watching. Thank you all Mindsetters for watching here. Mindset Learn Extra. So we have 10 minutes to go and we're going to do your questions, like try so quickly to do them. And I still have to announce the Curio winner as well. So Aslan, we have to run this race
time is
quick,
not on our
quick,
side.
quick,
So
quick.
guys, you got to be sharp with us now. We're going to move with as much as we can do for you in terms of the questions. So let's take one question on a phylogenetic tree. We've got a few, but we're going to take one. So start the phylogenetic tree below and answer the questions that follow. The names are not so clear here. They've given you the names, and I'll help you along as we go along. I want you to concentrate on the branching. Let me just make some marks so you know what I'm talking about. You know, I'm talking about the branching there, and so on. Okay. How many years ago did the Neanderthals split from the Homo sapiens? Right. So you won't be able to see that. There's the Neanderthal there, and when did it split from the human? Obvious there. It would be 0.55 million years ago, around this time here. In this time, there's the Homo sapiens. There's the Neanderthals. Good. You're just reading again. You don't need to study anything. You needed to read from the graph. According to the phylogenetic tree, what happened to the Neanderthals? Look at what's happening. Here's the Neanderthal. Is it going anywhere? It's a blind spot, dead end. So that means it would have become extinct. Right? Which group of modern humans came into existence between 0.18 and 0.2 million years ago? And that would be the Papuan. Now again, you can't see that. You see, we're talking about 0.18 and 0.2. 0.18 and 0.2, you're going to go here, you're going to go across there, and there's that guy, sorry about that, there's the guy there, and there's a name given there, Papuan. There, you can't see there, but that's what I'm saying. Important is not how we get it, what the answer is how to get it, and that's what I'm showing you in this diagram. Name two groups of Homo sapiens that will be very similar to each other from this one here. French and Han Chinese, again, you needed to see the diagram. Look at where they are. Let me just show you these ones here. Ah, come on. These ones here. Because they are the closest related. The others are further away from them. So those are the closest in that case. How many groups of modern human exist today? That would be 5. Name the continent on which Homo sapiens are believed to have evolved. That would be Africa. Again, this could be contentious because that is from the out of Africa hypothesis. Again, what is the out of Africa hypothesis? Exactly what this thing is saying, that the Homo sapiens have evolved from Africa. The multi-regional theory says differently. It says humans have developed all over the world and then they migrated and immigrated all over the show. Name two pieces of evidence that support the idea that Homo sapiens evolved on this continent here. That means what it's basically saying, two pieces of evidence to support the out of Africa hypothesis, and that would be fossils, mitochondrial DNA, and mutations on the Y chromosomes. Anything there, any two of those three. If you're explaining that one, fossils, the oldest fossils were found in Africa. This one, mitochondrial DNA, a mutation on the mitochondrial DNA, which is passed from mother to child, leads back to a female in Africa 120,000 years ago, who's known as African Eve. So that supports that idea. Mutations on the Y chromosomes can also be traced back to a male in Africa some 60,000 years ago. So that's the story. Can you see with this answer we've already explained the whole out of Africa hypothesis. I'm hoping you're winning with me, guys. Which group of modern humans shown on the diagram is most closely related to the common ancestor? That would be San. You've got to go back to that there, and you've got to look at the sand, look at the sand there, that's the sand, this one here, and the sand is there. From all the others, look at where others are. They're starting in this area here. He's starting closer to the common ancestor. So it's all about proximity in the diagram. How close are you in the diagram? Where did the branch take place? When did the branch? They can ask you, when did the branch take place? Which ones, who was the common ancestor of Homo this and Neanderthals? Homo this, I can't even read it myself anyway. The diagram is unclear. It would be this common ancestor here. What's the common ancestor of these guys? All of these guys. It will be this one here, and so on. Can you see what's happening? You've got to go back where the fork starts. That is the common ancestor for the ones after that. Good. I just wanted you to get an idea of that type of a question. Describe two other types of scientific evidence other than DNA similarities that scientists could use to prove evolutionary relationships between humans and primates. Other than DNA similarities, what other evidence do we use? We use, that was biochemistry. We can talk about protein synthesis. We can talk about biogeography. We can talk about, sorry about that, we can talk about anatomy. In other words, those famous things, homologous, analogous, and vestigial structures. These things also do that. And how these two, how do we explain this? Both involve similar structures. So then what the hell, why are we giving them different names? Because homologous, from there, arise from common ancestors, according to evolutionists. And analogous, they're similar structures, but they arise from different ancestors. So that one is common ancestry, homologous, and analogous not. Vestigial are structures that are found on our body or in an animal's body that seem to have no function. For example, the wisdom teeth, the muscles of our ear, appendix in our body. These things are said to be redundant in our bodies. So why do we have it? Evolutionists say we have it because our ancestors have it, and that is why it proves common ancestry. That's one of the other reasons there. Good. This question here deals with speciation. I'm going to skip this one here. I'm going to go to this one here. This one gave you a diagram of aloe in hot and dry conditions, and it was separated by a barrier, and the questions go as follows. Identify the type of speciation, obviously because it's a barrier, allopatric. Describe what has happened. Notice, even though this example is something you haven't seen in your life, look at the answer. There was a single population. There was gene flow. It got separated. Now there's no gene flow. Now each of them on different areas, they grow independently. One area is hot, the other one is cold. So what does that tell you? Different environments. And therefore natural sex selection takes place independently. And the organisms become genotypically and phenotypically different. Notice that the answer is what we've been singing about in all our evolution lessons. This is speciation, okay? Explain how biologists know that the species are different, because if they put in the same environment again, they will not interbreed. And that is the definition of species. The organisms must be able to interbreed. I want to look at one last thing. In an essay, discuss 6 differences in anatomical features between the hominins and the African apes and advantages of each and what have you. Again, here's an essay. How would you answer this essay? You can use points if you want to, as long as the points make sense. Now here, you have to talk about 6 differences. So you talk about the 6 differences between humans and other primates. And second part of it, advantage. So if you say, for example, humans are bipedal, and those were quadrupedal. How is bipedal an advantage to us? We can see, we got a better view, we can use our hands for other things and so on. A larger brain capacity has led us to develop language, technology, and the rest of it in that line as well. So we've done quite a bit of advancement in that. So you would develop your essay in that way. Now notice that they are, and I'm glad that this is left here in the sense that we can see that the mark allocation is given as following. Content is 17 marks, synthesis is 3. Now let's talk about that synthesis mark. The synthesis mark, 1 mark is given for relevance. Are you talking about what we're asking you, or are you talking about genetics in your answer? Second mark is given for logical flow. When you're explaining, are you jumping all over the show, or are you saying one point leads to the next, to the next, to the next? And the third mark is comprehensive.
That
Have
doesn't
you-
mean sentences, or does that mean
No,
just as long as it's
Yes.
it's
Comprehensive means you've answered the whole question.
okay, yeah.
Right? In other words, in this case, and this is an excellent example, there's two parts to this question.
Yeah.
One is for the differences, and one is for the advantages.
advantages.
If you only answer the one, then you're going to lose that one mark. And notice
okay.
that the one doesn't affect the other one. They stand on their own. And that's how you will get your marks in that sense there, right?
Perfect.
And even if you look at the old, if you look at others, you will see that they tell you that this is what's happening. If there's logical flow and what have you, nowhere do they say paragraphs. You do not have to put your essay in paragraphs. This is not an English essay or a history
But
essay.
the format must be right, right?
The format must
Yeah.
be more or less in order. You
Okay.
must try as far as possible to write solid, full sentences with paragraphs if you can.
Okay.
If you can't, you summarize it. If you're running out of time, you put your bullets and you write as much as you can in a time.
yeah.
But I must say, no life science learner in this country should say that they're running out of time.
Yeah.
No, the life science paper has enough time for you to complete the paper. So make sure that you get to the end, you've got enough time, you can check it. What we must do in the essay, however, is the way the question is asked, for example, is asking you for the 6 differences. Obviously, you can put the differences differently in the way you want to, but give that first and then the advantages. Or you can link that this is the difference and this is the advantage it has.
Yeah.
This is another difference, and this is the advantage there. So you're showing a progression and a logical flow in what you are doing. Don't write, for God's sake, do not write too much. The essay is only 17 marks. It's 1 mark per fact in life science. We don't give you 4 facts and 1 mark. 1 fact, 1 mark. A fact should go 1 line, 1.5 lines.
Yeah.
A page has about 22 lines.
So it should be about just a page. Even
A
that's
page
even
and a
too
half.
much.
About a page and a half.
Okay.
If you write big and you're spreading it out.
Yeah.
Page and a half, 2 pages maximum. This is a plea from all the markers in the whole country. Don't write 5 pages for 17 marks.
Really?
Because we've got to mark all of that for your essay. So please, guys, think about the markers itself and yourself. Why are you writing so much? If you're writing more than 2 pages, you are writing a lot of, you know what
Can
I'm
I
talking
announce the
about.
Curia winner
Please
before
do
you
that.
say goodbye? Well done to Mduduzi Tabede. You have won yourself 100 Rand airtime courtesy of Mindset. So Mduduzi Tabede. So thank you guys.
Well done.
Really, really hope you guys have good luck in your exams tomorrow.
No
Aslam?
luck, all the best, because
Yes.
exams about skill, not luck. So all the best to you guys. We hope you do well tomorrow, and tune in on Sunday at 3:30 where we do paper 2, guys. Lots of people
Awesome.
have problems with paper 2, so tune in on Sunday. We're going to take you through to get an ace in this paper. Do us proud.
Thank you, Aslam. Thank you, guys. Good luck. That's all from me to you. Cheers and goodbye.